Okay, good evening, everyone, and welcome to Understanding Ag series of webinars. This evening, the topic of our webinar is soil building secrets. And we're very pleased tonight to have our own Ray Archuleta and David Kleinsmith as presenters. So we will have co-presenters this evening. And we will start with Ray Archuleta. So in way of introduction, Ray spent a career, about a 30-year career with the USDA NRCS and became known as the soil guy. And Ray has been instrumental over the last couple of decades in helping people all over the US and many other parts of the world come to understand the value and the benefits of healthy soil and what it means to our farms, to our ranches, to our livelihoods, to our ecosystems, and to the quality of our life. Ray is an outstanding presenter and always does a tremendous job in helping people to be able to understand the nuances of the soil health principles and how to accomplish those on your individual farms and ranches. So Ray, I'm going to turn it over to you, sir, and let you begin.
Thank you, Alan. I appreciate that. Most people don't realize that I paid Alan a couple of bucks to say that wonderful introduction. Thank you, Alan. I appreciate that. Folks, I hope all of you are having a wonderful evening, and I'm glad you came to join us. We're living in some incredibly exciting times. Exciting in the fact that I see incredible hope in the future for agriculture. Years ago, I didn't have that kind of hope like Alan was talking about. I had 30 years within RCS. About 15, 16 years ago, I realized that I had missed the mark, that I wasn't very effective in my job. And a lot of it was the fact that in the points that I'm going to show you tonight is the three major points that I'm going to build for my slide set and my presentation. Is the first one was I didn't have a proper framework. So when I left college and I attended university for agronomy and soils, I didn't collect the proper framework. My thought process was fragmented and it wasn't put together. Nobody gave me a very holistic view on how to look at the natural system, and I'll go over that. The next one was I really didn't understand the goal, to be honest with you. I thought working with NRCS and getting a degree in agronomy and soils, that the goal was, and sadly to say, I thought was to help the producer with yield. And I'm going to talk about what the real goal is. And then the other part is I really didn't understand the context. And it's very frustrating when you don't have the right framework to look at things, don't have the right goal. Agriculture could be very frustrating. I started looking at agriculture like a burden. When I would come from work and work on my farm, it started to be a burden because I didn't know what I was doing. And then I also didn't understand what the context that I was dealing with. Next slide, please. Next, there you go, Alan.
Yeah.
As Alan is looking for that next slide, one of the things that I wanted to talk about is that what is amazing me when I go across the country is that most farmers, and myself included, we would look at the farm and the natural system, nature around us, as total separate entities. And I did that. When I left from school, I didn't realize that all these four ecosystem processes was occurring. As you see that slide that Ellen just brought up, the four ecosystem processes that we're going to show you in this slide happens everywhere, ladies and gentlemen, everywhere. we have diversity of life. And notice I put the diversity of life in the center. And the sun runs this. It's the energy, capturing this incredible light energy, these photons, and turns it into photosynthetic energy that feeds the diversity of life. And the reason diversity of life goes in the center, because it regulates the water cycle, it regulates the nutrient cycle. In fact, the diversity of life is how the natural system works. It transfers information through DNA, transfers nutrients, water. But I think a lot of us don't realize that the plant and the soil help regulate the water cycle. And without plants and soil, we can't have climate. And that is being missed when I was taught in school. And also the biogeochemical nutrient cycle. All these, as you can see, they're intimately linked. But it was astonishing to me, and myself included, that when I would talk to a producer, the first thing they would say to me, but Ray, you don't understand. Things are different here. We have different soils. We have a different climate. I beg to differ in the sense that life runs through all the planet. It's diversity that modifies and regulates our living planet. Go to the next, please. Next slide. Just to build my point even further. As Ellen is bringing up that next slide, this slide had when I make this slide, I read an amazing book from a Russian, and I can't remember his name now, but he was a brilliant scientist. And I never forgot this statement. It says, the greatest geological force on the planet is life itself. And I love showing this slide to landowners when I tell them. I said, now, landowners, that is a compaction issue. And they start to laugh. And I said, but how does that plant survive that an incredible restrictive environment? How can it modify and live and survive? The reality is that this tree has an incredible arbuscular mycorrhizae relationship with the roots, and the roots in that arbuscular mycorrhizae fungi can release these incredible enzymes and these incredible exudates that help break the rock down. And then when there's moisture in that water film in that rock, also with CO2 releasing from the roots, you also get carbonic acid. So the power of life to be able to break rock down. So the strongest, most powerful force is life itself. Next. As Ellen brings up that next slide, I know we're having a little lag time, but bear with us. This next slide was shared with me by a producer in Australia, and I thought it was one of the most beautiful slides, because look at that farm. A lot of times I hear producers say, well, look at the soil here. You know, we have really tough soils. Well, how about these producers in Australia? That's a gypsum seabed. And look how dark is on the top. Look how life, the power of life, the living root, biology, the microbes, were able to transform that top surface to get life. Next. So one of the things that I really think about how powerful regenerative agriculture and how I missed it in college. I didn't understand the power of relationship. So, oops. Yes. Oh, and so the power of relationship and understanding that the relationship of all these organisms working together in synchrony. And so one of the things that I started to realize, not only is life very powerful, but the goal, and remember I was telling you the goal in the beginning of the slide, said the goal is to mimic nature. If I would have had one professor, one person to tell me, Ray, your goal is to emulate nature, understand its synergies, its collaborative forces, understand all its synchronies, to love it, to nurture it, that would have been extremely helpful to me. It would have helped me help a lot of people in my career. When we show you this slide, we're talking about, because I've had producers say, well, Ray, do does it have to look like the forest and the prairie? No. The beauty about it is you mimic its architecture and its design. And that's the whole concept of a regenerative agriculture. If you understand the goal, to mimic nature, understand relationships, it is an incredibly powerful way to start going down the path of making your soil function. Next. One of the things that we were talking about, how we want to make our soil function. One of the things that most of us, we walk in the forest, and sometimes we kind of just, we're walking in passing, but sometimes we don't ask ourselves very deep questions like, why is that skin there? Why is that leaves there? What purpose do those leaves serve? Those leaves in the forest or in the prairie act as an incredible skin. They protect that soil. They feed the soil biology. We are now learning and helping producers not only plant these multi-species cover crops, as you see the picture on the left. Again, notice the architecture, see all the pollinators, see all the beauty, all of those leaves capturing energy differently. As you see in the picture, as we roll the cover crops, and David will talk about the logistics, but now we have producers that are actually emulating nature to this level as we are mimicking the architecture, but also mimicking the skin as we roll these cover crops, mimicking the forest floor and getting all the benefits of regulating the temperature, feed, and making it a habitat. That skin is also a habitat. Next, please. One of the most profound things I've learned from a researcher one day that I was reading, and I'll never forget, I was in the back of I was reading in my bedroom, and I never forgot how important it is to see that everything's connected. And I think if you realize how we talk about all these principles, like the principle of compounding, cascading effects, like Ellen talks about, there's no singular effect. And the reason for that is if you read this statement, learn how to see, realize that everything connects to everything else. One of the most profound things you can understand on your farming operation or your ranching operation, everything is connected. There's this beautiful web. Everything's connected. We're connected. Everything is connected. Why is this important from a practical perspective? The reason for that is if you don't understand, you don't understand the implications, what your tools can do and how they impact all of the living biota and all the living organisms. So the other thing that I thought that was a very profound thing that I've learned was the plant and soil are one. I personally think that we teach about soils incorrectly. The way I think about it is, if you do not have plants, you do not have soil microbes. If the plant is not feeding the soil microbes, then if you don't have soil microbes, you have geology. So without plant biology and soil biology, you have geology. Plants are amazing. They're the biological primers. Recent research has shown that these plants are also heterotrophic. They're also, they can eat, they can get their nutrients from eating other organisms and feeding on bacteria. They can take up organic nitrogen. They regulate the temperature. They are amazing. So without plants, this is the most powerful thing that we can understand as farmers and ranchers, is plants. Plants are powerful. Next. Why are these plants so amazing? This is one of my favorite little videos. This is a root leaking root exudates into the soil. Hundreds of compounds, liquid sun. A lot of those compounds, not only acid, proteins, sugars, all these bringing life into that incredible matrix of geology. Without this happening, there would be no life on the planet. This is an incredible thing. And one of the most important things farmers can do is plant it. I had one farmer tell me, he says, well, I don't have any money for fertilizer and chemicals. I said, plant it, and the biology and the plant together will cycle the nutrients. This right here shows that beautifully. Next slide, please. So one of the most important things, and once producers understand how soils function, is plant it. And this is what happens right away as soon as that root starts leaking this liquid sun. It starts to communicate with all these organisms. It starts feeding a myriad of these organisms. And then when those bacteria multiply at every 20 minutes, then the rest, protozoa, nematodes, all of them, you can get anywhere from 17 to 116 pounds of nitrogen just by this activity. In fact, we know now that 40% of organic matter is dead carcasses of this bacteria. Thank you, Ellen. The next slide. So one of the most incredible things to do on your operation is to plant. Never leave that ground bare, because the moment you leave that ground bare, this activity does not happen. Most people don't realize, and this is why I created this next slide. Please look at the root as an oasis in the desert of mineralogy. The soil is, all of our soils have this, sand, silts, and clays, whether you're in North Carolina, Tennessee, Kentucky, any part of the world, we all have sand, silt, and clay in different mixtures, different mineralogy. But the key here is I want you to look how the power of life. That's why your spacings between your corn and there's nothing growing in your plants, there is a desert. Where there is living roots, there is life. It's an oasis. That's why these grass systems, pasture systems, anything with fibrous root systems and that have a long root have an oasis, and they're feeding, and you have less and less of a desert of mineralogy. So the next slide, please. And to build my point is the next slide will show you a slide from my one of my friends of Adam Daugherty in Coffee County. And it will show you how powerful the root is. I'm thinking Alan's having some problem with that slide. There we go, Alan, thank you. And so at the left, you'll see conventional no-till, corn and soybean. This is in Coffee County in Tennessee. It has been in no-till for 20, 30 years. Most of 80% of that county is in no-till. And so it's corn and soybean. And you can see that stratified layer. You can see that that soil is not really healthy. But if you look at that picture, after 3 years of multi-species mixes, you could see that the respiration rates increased by 279%. The WEOC, more food consumption, more nutrient cycling, increased by 39%. And 50% increase in soil health score. It is absolutely amazing. So in the left, you can see the biggest complaints that most people had in the organic group or others that first tried no-till. Well, no-till doesn't work. We have stratification. You have all these issues with no-till. And we know that no-till obeys one of the principles of soil health, and it's part of biomimicry. But why does it fail? Well, we miss the other part, the living root. Notice how that root and biology absolutely transform to these beautiful aggregates. Those aggregates are these little cottage cheese, these little babies. That's habitat. That soil has completely changed from a not stratified. The left shows a stratified system, problems with porosity, with nutrient cycling. Look at the soil on the right, just 3 years of covers. Next. Not only is the roots incredible for making that habitat we critically need, but those aggregates actually help for infiltration. And the next slide that Alan's going to show is those aggregates create those beautiful symmetry. If you can see this incredibly video, the 3D model. our soils, and that model we just showed right there, that is what our soils are. They are a system of aggregates and cottage cheese formed together, creating these underground caverns that water flows. So the more aggregation, the more pores. And these microbes live in these beautiful colonies, as you can see. That is what our soils are, folks. And so, I guess I apologize. My slides are showing very slowly here in my internet, but they're showing appropriately everywhere else. So let's move on to the next slide. Thank you. Thank you, Gabe, for that. So this other is biodrilling. A lot of producers say, well, how can cover crops and microbes bring us nutrients? This is an incredible photo of taken by of biodrilling. Notice the scarring of that mineral, and that was done by mycorrhizae fungi. They did that. Again, those powerful enzymes working in synchrony with bacteria and mycorrhizae, they're the ones that bring the mineral out of the rock. That's why it's called biogeochemical nutrient cycling. Very powerful. This is how we can get away from a lot of our phosphorus, potassium, and a lot of our costly inputs by doing biodrilling, cover crops. Next. So I was, again, I apologize for my slow internet. There we go. A lot of times we talk about soil microbiology, but I wanted to make it very clear that when I read books, I try to search for books on soil ecology, not just soil microbiology. Why? The soil microbiology deals with everything on the top of that surface. You can see that on top of that soil profile. Notice those incredible links, those connections, very important. Soil ecology deals not only with the top and the surface, but the microbes below. Soil microbiology has a lot of focus with the microbes below, inside the porosphere. Please look at the collective whole. We need to understand that the top is so critical, just as critical is what's happening to the porosphere. So I just wanted to show you, when you look at the system, those systems are intimately connected. Next. So these incredible organisms make these incredible substances. I call them super molecules. Hit the next again, Ellen. And so what you'll see here is this incredible, and the picture here done by Dr. Chris Nichols shows geology, sand, silt, and clay. But when you add the activity of these organisms, these superbiotic glues created by the living roots, all these predation, all these organisms, this is what is different. Our management really dictates this part where we make these superbiotic glues. These superbiotic glues help regulate pH. They regulate water, moisture. These glues are absolutely important. So geology with these superbiotic glues is what you have is soil. Okay, so speeding it along here, the next slide I wanted to show you that is showing you is a manure pile from the Chihuahuan Desert. And if you can see that, that is absolutely incredible. Think about this, folks. That Chihuahuan Desert, less than 6 to 10 inches of rain, extreme temperatures. In fact, I went to college at New Mexico State, and it was right at the edge of the Chihuahuan Desert. Look how that manure pie is transforming that geological soil surface. Most people don't realize our deserts, that desert is really a prairie, and it became a desert because there's no more cycling. I wanted to show you this slide because how powerful animals are. Plants and animals are probably the most powerful things we can do to make our soils healthy and functioning. Next slide, please. And so, as you can see the next slide coming about, it shows you how the ranchers in the Chihuahuan Desert are getting the manures and any carbon source, any carbon skin. And notice the picture right next, what happens. Yes, even in an area with 6 to 10 inch precip, very hot temperatures. And I used to live there and again, attend college. The heat is incredible. And look at the incredible transformation. It is amazing what these organisms, the food and these biological messengers that excite the biology, and look what happens to the living plant. And the plant starts to manifest itself. Next. And so getting close to wrapping it up here, one of the things I want to say that I think that once we understand how the way the soil works, that it needs a living root and it needs animals in the system, then the self-regulating, self-healing, and self-organizing mechanism starts to kick in. But what hurts these mechanisms is what I call death by tools. It's the way we do our tillage, excessive tillage, the way we hay, excessive pesticides, excessive fertilizers. All these tools hurt the way the soil functions. We can inhibit soil function. So it's very, very critical how we use these tools. So, you know, a lot of times we get into the regenerative ag and we focus so much on the tools, but we're saying don't focus on the tools, focus on the understanding. This slide shows a perfect example of what happens when we do too much tillage and chemical fertilizer. And if you hit that slide done by Dr. Chris Nichols, if you focus and you look at that incredible complex web, you see the mycorrhizae. And then if you affect that organism, you affect the other types of organisms, and then the whole system collapses. And then the system no longer has the ability to self-regulate, self-heal, and self-organize. The next slide will show you a very powerful example of the acronym that I use. And you can find this in Dr. Stephen Gleason's book. It's really the frequency of disturbance, the intensity of disturbance, and the scale of disturbance, and the timing of disturbance. All these are critical. The whole concept here is you've got to limit the frequency of your tillage and your haying, or even leaving it fallow, and the intensity, the scale, and the timing. All these make an impact. That's why I use the acronym FIST. You can find this in Dr. Stephen Gleason's book on agroecology. Next. Those are the ones I already told you about, the self-healing, self-regulating, self-organizing, your body self-healing, self-organizing, self-regulating. The soil's the same way. Once you start bringing in diverse covers, diversity of animals, this mechanism starts to kick in. Next. And if you click on that, Ellen, some more, please, and you'll see the rest of the slide appear. This is a perfect example, and make sure that this is in South Carolina, and this will show you the soil on the left is Mr. Coleman, and Mr. Coleman has been corn, soybean, and wheat, and now they're using 15 species cash and cover crop. I mean a cash crop and a cover crop. No P, no potassium, no fertilizer, no lime, and you can see the soils on the left. The only difference that separates those two farms is that road. In the right, you can see that soil. It looks like the back end of a cow, no structure. You can see the left one, the organic matter increased in less than 4 years. Now, these are very sandy soils, folks. This is an area that gets 35 to 40 inches of rain. Very, very sandy soils. And look at the transformation of those soils. What's changed? Mr. Coleman changed. He understands the goal. Using the covers. Next. And here shows Dr. Busklude provided the slides. And here I want to show you each of those lines. If you look at the bottom of the axis is November 13th to April 16th, and you see the percent organic matter on the left, I just want to show you in these very, very sandy soils, and the only thing they've done in 3 years, you can see the upward trend of all those farms. Each of those lines, those color lines, represent a farm. Notice all of them in the sandy soil went up, just 3 years of covers. Next. The next one you'll see is the soil P. No phosphorus added in 29 months, and notice what happened to all those farms in very, very sandy soil. The amount of phosphorus increased by 15 pounds, and nothing was added to those farms. On the average, they increased 15 pounds to the acre. What happened? The living plants. That's what I'm saying, how powerful those living plants. They start the self-healing, self-organizing, self-regulating mechanisms. The next one slide will show you is the pH. And one of the things we're finding out more and more, that for many, many years as agronomists, we used to think that we added sulfur or we added gypsum, that was the best way to regulate pH. This slide will show you that all these farms, no lime added, and all the pH went up. The point here is to show you that, again, nature's self-healing, self-organizing, self-regulating. It's these organisms, it's the carbon, it's these functioning soils. On the next slide, we'll show you what I'm getting at, that in a soil that is in the left, and these two pictures show soils that were once the same. The picture on the left, most of us don't realize throughout the globe, most of our soils were degraded. This soil on the left is a forest soil. On the right soil is the same soil. The reason I want to show you is when the soil is like in the left, very well aggregated, high organic matter, high functioning system, pH is no longer a factor. pH is no longer a factor. It regulates itself. That's why it's called a resilient, highly buffered. It regulates its temperature, regulates its moisture. On the right, if you have a soil that you're still having to depend on pH to make all these nutrients available, it shows you that this system has low functioning biology, organic matter. It is not a functioning system. That system, the majority of our soils in this country still have to abide by the concepts of pH. Once your soils get healthy, that's no longer a factor to making your nutrients available. The last slide, please. Finally, I want to say, when you have the proper framework and you understand the power of life, you're always very careful to use the tools that promote life, plants and animals. So ask two things from us, really desires two things, plants and animals. And then we understand the relationship, how everything's connected, and we start seeing things as a whole. It really makes a difference in your life. Again, my goal now is I judge all research, all papers that when I'm studying, all talks, even myself, on the goal. Biomimicry, mimic life. My goal, our goals, is to encourage relationship, life, and then the last one is understand your context. And David's going to talk about how you can facilitate the logistics in your context, no matter where you're at, and that you need to acquire the right proper mindset, learn the skills, and know the difference between tools, skills, and the mindset. And last word I want to say to you, when I travel the whole country, ladies and gentlemen, and I come across a lot of producers, one of the most important the most important thing you can learn, that I have learned, it's about the mindset. Farming is about your mindset, having the right goal, the right mindset, and understanding the collective whole. So David, I'm going to turn it over to you so that you can lead him towards how do they facilitate and carry out the logistics.
Thank you, Ray. If you want to go ahead and click next, we're going to talk a little bit about using cover crops and livestock to how to build our soils and soil health. And one of the first things we need to focus on is soil health first. Click next, please. So we want to utilize principles and not practices. And there's a lot of practices out there that people are talking about in the last, really have gotten a lot of interest in the last year, and, such as interseeding and wide row corn and stuff like that. And those are practices that are extremely useful and valuable, but we have to understand why we're wanting to do them and understand, kind of some basics behind how to utilize those on our farms. But really we need to look at, the principles of soil health. They're the principles for a reason. And, I put context there as the first one, just because it's about the understanding of, why we want to use a cover crop or why we want soil health. Understand how we're going to graze them, if we're going to graze them, understand what a interseeding will potentially provide for us as well. But we've talked in previous webinars about the principles, so we don't have to go into them in detail. But it's always good to reiterate them. Minimizing soil disturbance, keeping the soil covered at all times, adding diversity into the cropping rotations and our cover crop mixes, keeping a living root in the soil as long as possible, and integrating livestock. Next. So it all starts off first with, we have to have plants in the system. Our first trophic level is that photosynthesizer. So we have to have carbon entering into the soil profile through living roots. Having ground sitting fallow 8 months out of the year does not provide us any food for our biology. And so as Ray was talking earlier about, if we take one thing, it's kind of like everything's connected with a string. If we pull on one thing, everything falls apart. So if we take those roots out of that system or living plants, we start to reduce our organic matter. We don't have the food for the biology, for the fungi. Our coyotes of the soil, our nematodes and protozoa that feed on bacteria and fungi, they aren't there and cycling, so we're not able to cycle those nutrients. And so it's, as Alan mentioned in previous webinars, it's a cascading and compounding effect. Next. So one of the things when I was in ag retail and worked in sales in a number of years, we always focused on this type of model on driving yield. And we always looked at, okay, if we're doing tissue sampling or pulling soil samples, we're looking at what's our most limiting factor there, and that's going to really basically is going to be our yield drag, right? So the number one thing that this whole thing is missing is carbon and the biology. This was solely focused on a chemical system, and that's how we got it all so wrong, is we took the biology out of it and we gave the living roots of what's the carbon being pumped into the soil system no value at all, and that's what cycles everything. Next. So again, carbon is the key driver. Carbon is basically that food source that keeps everything rolling. There's some really good YouTubes out there, and Keith Burns does a really good job of talking about carbonomics and how important carbon is in that soil economy, and how that carbon helps to cycle nutrients and increase water infiltration and retain water. But it's kind of that stimulus check, if you will, for the biology always. Next. You want to hit play on that? So the interesting thing is on this video here, it's going to show, this is a ditch here where it's really easy to push that spade in the ground. And the one thing that a ditch always has is living roots, and always has that skin or that armor on the soil surface. So even right here, I'm really close to that wheat field, but it's really incredibly easy to put that spade in the ground. And we get here, And you can notice I struggle a little bit, actually a lot. I can put my whole weight on there and not get any deeper than probably 2 inches into the soil profile. But throughout that field, anytime that there was any type of heavy residue with corn stalks, I could kick the corn stalks over and easily push that spade into the soil profile with wheat right next to it. So it's not the wheat that was sucking up the moisture and making it so the soil was hard, but it was the management practices before. So diversity is huge in building our soil profile and feeding diverse microbes in our soils, but keeping that soil surface covered at all times is absolutely essential. Next. Oh, next. So one of the things, some of the considerations for utilizing cover crops and having success with cover crops, we need to ask ourselves some questions. And right now, being the spring of the year is right when a lot of guys are getting ready to plant their corn or soybeans, milo, sunflowers, cotton, whatever. We need to be kind of thinking about these things. If we're going to have cover crops into our rotation, we need to have these in the back of our mind of what are our goals that we want to achieve with those cover crops. What type of residual herbicide program am I using, and what type of carryover will I have? What's the next cash crop? What's your annual rainfall environment? When's your first killing frost? That's a big thing, you know, when you're looking at seeing diverse mixes and you're looking at when your harvest will occur. If you're planting, let's say 120-day corn in central Illinois and you're delayed harvest now until mid to late October, well, the chances of actually getting some species to germinate and survive is pretty limited. So how will you seed that cover crop? What are your termination options? And will it be grazed or not? That can also have an implication on that herbicide carryover too. There's restrictions on herbicide labels for grazing. And really it's very important to watch this if you're grazing any dairies. But some of the other questions I kind of like to rank when I'm talking to guys about cover crops is, what do you want your cover crops to provide to you? You know, and I have them list them as a numerical order instead of just having to check the box to understand what is most important to them. You know, it might be to produce nitrogen, to increase organic matter, reduce erosion, improve soil structure, provide weed management, cycle nutrients, improve soil moisture, feeding livestock, improve diversity. Maybe it's pollinators, or even managing soil-borne pathogens. Next. So really, when we're really sitting here and thinking about our cover crops and thinking about our next cash crop that we're going to seed, so if it's the fall of the year and we're getting ready to seed a cover crop that's going into corn, let's say, we got to really understand this carbon to nitrogen ratios so that we're not limiting our corn crop by having a cover crop out there that is going to tie up nutrients for too long. So we need to understand that the ideal microbial diet is at a 24-to-1. What that means is that microbes really need about an 8 parts carbon and 1 part nitrogen for just maintenance needs. Then they need another 16 parts carbon for energy. So, you know, if we're just sit around on the couch all day, we don't require a whole lot of extra nutrients or anything. We can get by with pretty minimal, right, for maintenance. But if we're out and doing a lot of manual labor and everything, well, we need a little bit more energy, right? So that's where the soil biology is the same thing. So then we have to understand that when we get above that 24-to-1, the cover crops begin to slowly decompose. So, you know, typically, for me, I tend to like a carbon to nitrogen ratio in front of corn to be somewhere between a 30-to-1 up towards a 40-to-1. But, you know, if we were just to go out there, let's say we're going to go and plant soybeans into a cover crop, and we used a mixture that had maybe some hairy vetch in there and annual ryegrass, for instance. Well, that seed-to-N ratio is pretty low. Annual ryegrass isn't listed on this one, but annual ryegrass can tend to be around 17-to-1. So it's going to cycle really quickly and not leave a lot of residue on that soil surface. So, you know, you're not going to have that weed suppression as if you would have planted a cereal rye and let it go all the way to past anthesis. You could have residue that lasts out there until harvest or after, depending on how active your biology is in your soil profile as well. So this is really important to understand this chart to know, okay, what am I going to plant next? And how can I benefit by getting those nutrients out of that residue and into my next cash crop? Or am I going to keep that residue on the soil surface longer to keep the soil cooler, maintain soil moisture, and suppress out weeds? Next. So when we're talking about, you know, right now is a big time when everybody's thinking about, you know, what chemical applications you're going to be applying to their cash crop. And we really need to understand this is some work from University of Missouri, and there's a whole lot of good information out there on the internet, and I believe we're going to be sharing some of those on the YouTube show notes page, basically. Some links that you can go to find out some of these carryover issues for different herbicides to different cover crop species. You know, the biggest thing is, you know, for soybeans, you know, cereal rye is pretty resilient, and even annual ryegrass to most things. But if we were to use like Zidua at 3 ounces, and we were planning on planting annual ryegrass after soybean harvest, where you're looking at more than 30% reduction there in your stand. That is that's an eye-opening number to me that I would look at and say, okay, well, maybe I want to use, instead of Zidua, maybe I want to use dual. Because, you know, either I'm going to have to increase my rates to offset the chemical injury, carryover injury, or I just use a different herbicide method and I'll be okay. Next. And the same thing with corn as well. And one thing that sticks out to me on both of those is the Austrian winter peas there. You know, if you go back and look at that slide and look at this one, most herbicides have a pretty damaging effect to Austrian winter peas, you know, 15 to 30% reduction. So, you know, if you're planting 8 pounds an acre of an Austrian winter pea, you know, at 30% reduction, you're going to look at, you know, about 2.5 pounds roughly loss there. So, you know, that's money out of your pocket you just lost. So basically, instead of getting 8 pounds, you're only getting about 5.5 pounds. Oats are another thing, you know, you look at the percent reduction there. A lot of times oats are in a lot of cover crop mixes, and you start looking at that list of herbicides and some of the carryovers, boy, there's a lot of them there that can really tie some things up. Next. So the other thing to be cautious of, and this is really important even on interseeding, is this herbicide residual half-life. And what this basically means is the half-life is the time required for 50% of that chemical herbicide to break down to secondary compounds. So, you know, if you're looking at using a product like sulfentrazone or Authority, depending on your soil pH, the amount of rainfall that you have, the texture of your soils, and where you're at in the country, basically, you can be looking at anywhere from 32 days to 302 days. You know, that can have a significant impact on the cover crop that you're going to seed out there to having any type of success. So just be cautious of what we're doing out there as we're making our decisions for the year. Next. So one of the greatest things we need to focus on is how do we build a soil aggregate? Once we build a soil aggregate, so building the soil aggregate is more important than probably anything in soil, in my opinion, just because when we build a soil aggregate, we build that the microbial habitat, we're feeding the microbes in the soil more, we're able to store more water and cycle more nutrients. So if we look at the typical corn and soybean rotation, we plant corn in sometime in April or May, so comes up in May, let's say, and we harvest sometime maybe September, October. Well, if you really start to look at how long that corn plant is actually growing vegetatively and releasing root exudates, it's really only until from probably May, 1st of May, depending on when you're planting, till about somewhere in the first 15 days, I'll say, of August. And soybeans are about the same. Once you get into reproductive stages on those two crops, you're not releasing a lot of root exudates, so you're not feeding a lot of biology. So really, when you think about it, that corn or soybean crop is really only feeding the biology, you know, 3.5, maybe 4 months out of the year. So if we were to think about our dinner tonight, if you had a cheeseburger for dinner tonight, and that's the last meal you're going to get for the next 9 months, boy, you would be a bear to live around. I can tell you that for a while, and you're going to get pretty hungry. And that's what we're doing to our soil biology. So we can look at, okay, if we're going to put in a cover crop, we can put one in. We could either fly one in into a standing cover crop, or we can wait until after that cover crop is harvested, and then we can drill in a cover crop in that September, October timeframe, and it'll last all the way up until we get ready to plant the next cash crop again. Next. Thank you. So really, if we want to look at how we can diversify our cropping systems and utilize more like some of those practices I was talking about of interseeding, we can look at, you know, planting corn in that April-May timeframe, and by the time that corn's about V6, then we come in there, or before, depending on your environment, I'll say that, depending on your environment, you can go in there up till V6 and seed a cover crop to grow with your cash crop. So this has got some tremendous opportunities, especially if you're planning to graze, or if you're in a northern environment where you have a short growing season, then you can get more cover crops growing. But if you think about, you know, this August time frame when that corn plant's starting to shut down, and you have a cover crop there, well, that cover crop is still going to, even though it's basically semi-dormant, it's still feeding the biology. And as soon as that canopy opens up and that cover crop and the interseed takes off, it is feeding the biology. It's kind of like a trade-off. You know, it's feeding the biology like crazy. So looking at some of the things that we can do you know, we need to look at our fields and think, how can we go from, how can we increase our rotation basically? And what benefits would that give me? So, you know, we start out with corn, we do the interseeding, then we can go into soybeans, and then after soybeans, let's say we go to a wheat or a small grain, and with that, you know, the next April we could go in and we could, or February, March timeframe, we could frost seed some clover into that, harvest our grain. We've got something already there growing, and as soon as we harvest our small grain, we could go in there with a warm season, big biomass cover crop to feed the biology an insane amount of root exudates. We follow that back up. Maybe we can either mix in a cool season into that warm season, or we wait and then maybe we drill in a cool season in September again into that warm season. it would follow it up with maybe corn again and start the whole cycle over. Next. So interseeding covers is a really good resource if you're thinking about interseeding cover crops and looking at the stages of plant growth of when you need to be interseeding covers. You know, so really you don't want to interseed cover crops into corn at the same time that you're planting corn. Corn's kind of a little bit of a weakling. It doesn't like a whole lot of competition as it's first growing. But if you wait until, you know, that corn's about V2, V3 time frame and start there and go up to V6, that's a good opportunity, or coming in later and either flying on or using a high clearance machine like a Miller or Hagee and dropping cover crops in between that row. Alan, if you could hit next, please. Again. So in that August, September time frame, you could then be in there to seed that cover crop with a high clearance machine or aerial application. Next. So again, I talked about those herbicides being important. If you're thinking about interseeding cover crops, this becomes really important to knowing, you know, what can and can't you use. Your options get pretty limited. So what I tend to like to tell people, you know, especially when I'm telling them to focus on your principles first, is to get a cover crop established after you harvest your cash crop the year before. Focus on trying to get that cover crop established. Just do what you can manage, and to make sure that you have enough cover crop out there that you can use that for some weed suppression, so you don't have to rely on the residuals as much. The interseedingcovers.com website has got a lot of good information on there about, you know, which herbicides work and don't work for interseeding. Next. Some of the interseeding cover crops that you can use are annual ryegrass, crimson clover, red clover, cowpeas, hairy vetch, radish, buckwheat, flax, oats, mung bean, sun hemp, and potentially many others. When we get into the northern climates, that's where cereal rye, oats, triticale, and wheat has a play there. But in the southern environments, it just doesn't work real well. Next. So as we can see, this is a kind of a map where the green shaded areas is where the interseeding cover crops has been really successful. If you'd hit next, please. That yellow line, north of that yellow line, tends to be about the area where maybe the cereal rye, interseeding cereal rye, or maybe triticale or wheat or barley might work better. That northern part of Illinois there, and that could be hit and miss. So that line might need to be moved up a little bit further north, but the you know, the opportunities there are pretty good to be able to use those. You know, if you use a cereal rye south of there, especially if you get down to about the central part of the state in Illinois, you know, across there and on Interstate 72 or so that goes through Springfield, Quincy to Springfield to Champaign, your chances of using cereal rye and getting it to last through that summer is going to be pretty slim to none. And it's going to be, quite frankly, a waste of money. And with the commodity prices right now, we can't afford to be foolish with our money on these trials. Next. So some of the equipment options that we can use for interseeding, you know, there's some guys that are rigging up like a Valmar or a Gandy type box on their side dress application and seeding that way. Or, you know, Don has the Don Duo units that you're seeing in the top middle picture. Being able to go in there and seed cover crops that way. Some guys are taking old 3-point hitch drills and just removing those row units where their corn rows would be, and doing it that way, which is a nice cheap way. Some guys are taking some old rotary hoe units and mounting a an airflow system on there. Other guys are using, well, it's not really interseeding, but a way to get a cover crop established, I should say, after harvest, because, you know, either labor restrictions or whatever, but mounting either a Gandy box on the head of a combine, or using, like in that bottom picture, a Valmar or something mounted on the backside to seed right as you're harvesting. Of course, going in there with a Hagee or a Miller or some type of high clearance machine and seeding right before harvest works really good too. It's probably, when you're doing that method or flying on, you really have to be watching, you know, what your soil conditions are like, how dry it is, and also what the rain forecast looks like. If you're doing it with no chance of rain out there, you know, again, could be wasting some money. Next. So this is the value of interseeding cover crops. Right at harvest time, you could be looking at a field like this within just a couple weeks, or even right at harvest, of a solid mat of green. So think about building that aggregate, and it's feeding that carbon into that soil at all times. So think about all of that green matter out there just feeding the biology, but the biggest opportunity to make some really nice money, next, is to put some cows out there. You know, that's what this field is missing right here, in my opinion. You got a tremendous opportunity when you're doing either interseeding or these wide row corn, is to get the cattle out there. You know, whenever we graze cattle, and especially whenever we're doing anth grazing, you know, and grazing adaptively across fields, we can significantly improve the soil carbon and the soil water holding capacity, the nutrient cycling, and the biology is in there as well. You know, it doesn't take a whole lot of time, you know, to set up a paddock design in fields like this, and to move cows on a daily basis either. But this is probably the most critical point. And I think, you know, when we look at putting up hay all summer long and feeding hay all winter, well, cow's got 4 legs and a mouth, and she does a pretty good job of spreading her own manure. So we don't need to go out there and be as labor intensive on a daily basis when it might be 20 degrees out there and she's happy as can be out there. Next. So with the wide row corn, you know, I talked a little bit about this 60-inch corn. This is a tremendous opportunity. If you look at the amount of biomass that's grown there, you know, we can see that, you know, there's a lot of fodder there that we could be using in grazing come harvest time. If you don't have livestock, or if you don't have a neighbor or somebody that you can rent your field to graze this, I probably would shy away from it. You know, it's good to experiment and to try a few things on some small acres and all, but if you don't have the livestock, I'd say there's no real benefit to doing 60-inch corn, unless you are just really wanting to jumpstart the biology a lot in that field. It's awesome for that, but you know, I think you could focus on some, you know, 30-inch or maybe even 38-inch corn and do interseeding in that and get along a lot better. Next. So I haven't done any wide row corn, but some of the things that I kind of think about is when I used to place hybrids, I would always think about the roots of those hybrids and how they fit in on a high population or low population, but also the structure of those roots for the different types of soils. So when you're looking at, this is some information for that Winfield is produced through their answer plot system, and looking at the response to low response to population hybrid, when you're pushing that up to, you know, let's say that low is at 30,000, and we increase it up to another 18,000 on top of that, up to, you know, 48,000, that root system really shrinks up. Where when we look at something that has a high response to population, it maintains that same type of root system as we get even those higher populations. So, you know, I don't know if that's something that would play into effect with wide row corn or not, but it's just something in the back of my mind that if I go to start playing with it here this next year, that I'm going to be focused on what hybrids can handle that competitiveness of being right close next to each other. Because when you're going 60-inch corn, if you typically would plant 30,000, you probably need to be in row at 60,000. So that puts those stalks really, really close together, and you don't want a little spindly stalk and have an August wind come through and lay everything down. Next. So diversifying the rotation, you know, one of the things I talked about earlier was, you know, putting a small grain in there. This happened to be a picture from Prevent Plant from this past year. This was an 8-way cocktail mix that was sown in July. So it had been about the same time as a big biomass cover crop would have been planted after wheat. And this picture would have been taken in I think it's the latter part of September. So you can see how much biomass we could grow there. And it looks like we rolled it down with a roller crimper, but actually the farmer just went in there with just a drill and drilled in a cool season cocktail mix right into that, laid down that warm season stuff, which really helped to armor that soil. We've been kind of wet in that part of the world all winter long, and it significantly reduced the amount of erosion there. And this is a field that I'm really interested in seeing, because if we look at the next slide, about 2 weeks before that cover crop was laid down, I went out and took a 2x2 square and cut the cover crop off at the ground level and sent it off to a lab to see how much nutrients were in there. You know, in a 2x2 square, there was 7.8 tons of dry matter per acre. Think about that. You know, if you're going to graze cows on that, that's a lot of dry matter. But, you know, it's a lot of carbon. When we look at the total percent carbon, or the total carbon per acre, you're looking at about 6,000 pounds. And the total nitrogen was at 279 pounds. So I put it right about a 26 to 1 ratio right there. So that residue actually broke down pretty quick. Looking out there here this past about 3 weeks ago, that residue is getting pretty thin out there over the winter time, just because of the mild winter that we have had. But the interesting thing is that, you know, we didn't fertilize this field at all. It hadn't been fertilized since the previous year, but yet that cover crop had nearly 280 pounds of nitrogen, 146 pounds of P2O5, and 654 pounds of K2O. Now that's more than triple the values on the soil test. Alan, if you'd hit next. So if we were to replace those nutrients, everybody always wants to talk about how expensive a cover crop is. Go back one, Alan, please. If we were to talk, everybody wants to talk about how expensive a cover crop is. If we were to look at replacing those nutrients that were in that cover crop with commodity fertilizer, well, using this spring prices, it would have taken 340 pounds of anhydrous at $89, 317 pounds of 046O at $57, 1,090 pounds of potash for $196, and a total of $343 per acre for a $25 cover crop mix. That's pretty reasonable. Next. So where did those nutrients come from? Well, it all starts with that little red circle was supposed to be around the aggregate formation, but it all starts with building that aggregate. Because once we build the aggregate, we improve the water flow, we improve the water storage, the water infiltration rates, we decompose the residues faster, we go around this whole thing, we cycle nutrients. And I think, you know, as we're looking at our commodities and everything right now, we have really got to be figuring out, how can we cycle nutrients? How can we store more carbon? How can we do these things and not have to write out so many checks? You know, Gabe always asks, would you rather sign the front of the check or the back of the check? Well, I'm kind of like Gabe. I'm pretty greedy, and I'd rather sign the back of the check anytime that I can. Next. So with that, we'll just open it up to any questions.
Okay, thank you, David, and thank you, Ray. So we will start with the questions this evening. Our first question is the soil health calculation or score. How is it calculated? Ray, do you want to start with that?
Yes, I do. Thank you, Alan. Okay, the soil health calculation is based on 7 parameters. The first one I like to think about is the water extractable soluble organic carbon. Notice I use that key word, water. Soils are subaquatic and they run on this labile carbon. It's the food as it feeds the microbes. And so it's based on that. It's based on CO2 respiration. The concept that Dr. Haney came from that when Rick and I were talking about that is when the Russians knew a long time ago that as you had more carbon and had more nutrients in that soil, the CO2 respiration went up. That is well established in the early 1900s. So, and it's also percent MAC, the efficiency of the microbes to process the carbon, which affects the respiration, organic nitrogen ratio to the inorganic nitrogen, and the organic carbon ratio to organic nitrogen ratio. And then also the extractants Dr. Haney uses concepts of biomimicry. He uses citric, malic, and acylic acid as the root. He says, well, if the plant exudes, we want to mimic the acids, what the plant roots exude, not these caustic acids that are used in typical labs. So the Haney test is based on, it's a biological test, and it's not a chemistry-based test. So those are 2 different tests. And so that's why a lot of people have a hard time understanding the Haney test. You have to have a biological framework. And so it's based on those parameters.
Thank you, Ray. David, anything to add?
No, Ray did a great job covering that.
Okay, we're going to go to the next question. And Ray, this is asking for you. Ray, can you explain why a soil in a natural system can exist without any external fertilizer inputs, and how you can establish this on your farm and obtain good crop yields without fertilizers, just like Gabe Brown's farm?
Thank you. I think that really asked that question. Excellent question. The reason it does, if we need to go back millennial, depending on what you believe in, but these soils have been self-regulating, self-organizing, they contain themselves. They have been doing this for a long time, and it's biology. It's these complex webs that David and I were talking about, how they feed on each other and how the roots, in fact, research has shown, is that the plants, we're finding out that plants are much more intelligent than we've ever imagined. Is it plants, we know that plants communicate to microbes. Who's regulating who? Is it the microbes regulating the plants, or is it the plants regulating the microbes? We still doesn't know. There's so much complexity with it. But the point is, these functioning ecosystems were working quite well before we came into play. And so what happens is that when you diminish the system, you break those chains, those webs, you diminish the system. It's the way we use these tools. That's why if you watch how Gabe did it, and you understand how Gabe's story, he started because he didn't have these costly inputs. So he had to learn, and as he started building his soil, he started realizing, as he started becoming a farmer scientist, he started checking and started doing in his own testing that he didn't need a lot of these inputs. And he started just weaning himself slowly, but also coming across the right people like Dr. Chris Nichols. And many of us have come to the conclusion that these systems are self-organizing, self-healing, and self-regulating.
Gabe, since Willie also mentions you here, do you have any additional comments?
The only thing I will say is the sixth principle that was recently added is that of context. One of the things I'm seeing occur over and over all over the world is we are farming and ranching out of context. We're trying to outproduce our environment. One of the ways that we can really make sure that the system functions properly is by not trying to outproduce our environment. We can't tax it too much. It goes back to what many of us are saying. We will take profit over yield any day. And that requires a shift in mindset. We have to realize it's not all about yield, it's more about the profit. And when we allow the system then to dictate the yields, then we become much more profitable.
Okay, David, anything to add?
No, just like that image with that big biomass cover crop, those nutrients came from somewhere. So they're in the soil profile already, and the microbes are there too. Even though we might run a PLFA test and the microbes, the cyclers, the protozoa, and the mycorrhizae might be low, as soon as we plant the seed and plant the diversity and let it do its thing and let it get some size to it and pump the carbon into the soil profile, we start seeing these nutrients come up. I mean, it's all just part of the system and the cycle.
Okay, so Willie has a follow-up question to that, and it is, so crop removal replacement is not necessary in this instance?
Yeah, so you really have to be careful with that. Some people will try to cut cold turkey, and they're just doing corn, soybeans, and let's say, and they're not doing a cover crop, or they're pretty immature into a cover crop, like maybe the first year or two. You can start weaning yourself back, but don't cut cold turkey. There can be some significant harmful effects from that. Utilize the Haney test to understand how much nitrogen you actually have in your soil profile, or phosphorus as well, and go off of that and start backing it off. That'd be my recommendations.
Thank you, David. Ray, anything to add to that?
Yes, I think one of the things we have to realize, and I think Gabe did a beautiful job explaining that last comment he did, it's about context and what kind of type of system. If you're doing silage or hay removal, you're exceeding the ability of the biogeochemical nutrient cycle to keep up. And so if you think about, if you're removing everything, yes, that becomes an issue. The system can't keep up with you or what you're doing because you're removing everything. If you just take the grain, that's different from taking the whole stalk and everything with you. So you can exceed the ability of that context to be able to cycle quickly. So you do have to take that into consideration. Again, context.
Okay. Gabe or Shane, anything to add?
No, I don't have anything.
Okay. We'll move to the next question. What if your problem is high pH soil, say an 8.2 pH, and nutrients like phosphorus get tied up and unavailable for plant use?
I'll start off with that, Alan, because I grew up in an arid environment, and we experience high pH all the time, especially with sodic saline soils in the desert. And if you, again, go to the context again, a lot of those systems have been so degraded for so many years, and there has not been enough carbon flow. This is the result. If you saw that picture from the picture of Chihuahua, all those soils had high pHs. What happened? The lack of carbon flow. The first thing we start to do to remediate sodic saline soils in irrigation systems or any of those type of systems, we add carbon. We start putting cover crops. We back up on the fertilizer, back off on the tillage. We back off. So we add the nurse, cover crop, go to root-till. All of this whole system start change, and the pH starts to lower. Manures and carbon, all these systems, these super molecules, all these other things working in synchrony, you start changing it and lowering the pH, and then phosphorus starts to become available. A lot of this is because of the context where it's at, and most of the time it's very degraded. So we were able to remediate a lot of soils through a lot of those practices following the 6 principles.
David, anything to add?
No, as you mentioned, that slide of Alejandro's that Ray had, having the carbon there is huge, and feeding the biology. Yeah, Ray did a good job.
And I just want to add one more point is a lot of us don't realize that microbes can change the pH right around them. They can change a whole unit, and it can be different from one aggregate to another aggregate. So I think it's, we need to realize that even the concept of pH, I'll talk about that later, is probably not the right way we're measuring it. That wasn't the original concept of the way pH was supposed to be done by the physiologist and the mathematician who came up with the Sorensen. I think we took his concepts and tried to apply it to a soil, and it should only have been applied, pH, the concept pH should only have been applied to a solution like the ocean or the blood. So I think we've taken that concept and misapplied it.
Okay. The next question, Ray, when you were referring to the pH regulated slide, has there been a real documentation of the process used to achieve the result of the example on the left? And he's looking for a more specific process if it has been documented.
Well, there is, I think a person that's done something very fascinating is Dr. Will Brinton. If you go to Woods End Lab, he's got a publication, and he wrote a really neat publication about the dynamics, the new emerging view about pH. In fact, I learned a lot reading his paper and how he's guiding people down this, this concept that we may be way off. A lot of times when we applied pH, pH was only intended to be applied in a solution. And with soil, you pick up a lot of the mineralogy, and so we're not getting really true values of what real pH means, especially the measurement of hydrogen ions. So if you go onto his publication and there he wrote up a really nice, the new emerging view about pH. I think it's very, very interesting. But myself, I came to this conclusion in about 3 or 4 or 5 years, just going in the field and bringing all the pieces together, reading ecology books and all these other things. I started realizing we may not have this concept correct, in my opinion.
Thank you, Ray. Anything to add, David or Gabe or Shane?
I don't have anything.
Okay. All right, the next question, and David, I'm going to start with you on this one. The 24:1 C to N ratio comes from where? What's the research data? Who?
So I couldn't tell you, I guess, who did the research, but if you look at just Google ideal microbial, the soil microbial diet, it'll come up. It's on the USDA's website and everything else. So I guess if we're going to argue with anybody, we can argue with the USDA on a lot of things here. But there's a lot of research that looks at that ideal microbe diet. And also when you're looking at microbes as a whole, realize that bacteria and nematodes and protozoa and fungi, they all have different carbon to nitrogen ratios within their body as a whole. So it's kind of like an average of everything, but an average of what the ideal soil microbe diet would be. Because we know if we take a low carbon to nitrogen crop like alfalfa, and we send it off to the lab, we figure out what the carbon to nitrogen ratio is on there. And if we lay it down, it's on the ground and it's gone in a short amount of time, we understand that, you know, we don't have to have a fancy test to really tell us that, you know, it breaks down really quick. Where if we lay down something that has a higher carbon to nitrogen ratio, even, you know, cereal rye that's, you know, at boot stage, and it takes longer for it to break down, we can start to identify that these things are occurring because of that carbon to nitrogen ratio, basically.
Okay, Ray, anything to add?
Yes, it's on page 537 in the Nature and Properties of Soils, and in the 15th edition. David's right on. It's on page 537. That's Ray, and it's called The Nature and Properties of Soil. That is the 15th edition, and all universities use this as the major textbook. Dr. Ray Weil and Dr. Brady, page 537, and it'll tell you exactly what David said.
Excellent. Thanks, Ray. All right, so the next question is, what is the compaction risk by putting cattle onto cropland? I understand the benefits, but I worry about doing more harm than good in thaw periods. David, I'm going to start with you on that one.
Boy, we hear this question a lot. You know, the compaction issue actually comes back to, well, oftentimes it's a man-made issue. It's how are we grazing those cows? And granted, you know, Alan, in your environment, in my area this winter, it's been wet. You know, we've had an excessive amount of rainfall, and yeah, you can get some pugging issues and you can get some compaction issues if you conventionally graze cattle out there and just turn them out there and let them do their thing. Basically Columbus graze, you know. They're going to start creating paths from one end of the field to the other back to the water. They're going to pug up around the water. Well, if we instead we do amp grazing, and we do, you know, what you've mentioned it before as a tickle graze, and we just move them across the field really quick and just let them just take the tips off, we don't see those compaction issues nearly as much. And a lot of those, you know, it looks bad and there'll be some pugging issues out there. Well, if it's pugged down maybe 2 inches or so, I don't worry about that as much. No-till right into it, slow the tractor down, slow the planter down, adjust your seeding depth maybe a little bit deeper, and just plant. It'll plant just fine. You'll actually be surprised that you'll actually have some better crops than what you might think. Now, realize it's not the first step in that hoof mark that causes the compaction, but it's more like the 8th and the 10th and the 12th, maybe the 20th step in that same hoof track that causes that compaction there.
Yeah, and I would add, David, first of all, you're absolutely correct. It is definitely has a lot more to do with the way we manage that grazing than anything else. But I would also add that as we continue to build biology and therefore build the subsequent soil aggregate, then these issues become less and less and less of a problem. So as you build more aggregate, you simply can stop worrying about much of this. And Gabe, I mean, you in North Dakota, you deal with a lot of these issues as well. So I'm going to ask if you have anything to add.
No, nothing to add. Compaction, you're right, it is a function of time. And the other thing, just got to use good management. When it comes to that time of the year, when you're getting a lot of thawing and spring moisture, you need to move the livestock off to a sacrifice area.
We got to realize that sometimes the most dense compaction is right here between our ears.
Okay.
Hey,
Next.
Ellen?
Yes.
Ellen?
Yes,
I
right.
just want to add to that. I had some personal experience this. We were about 15, 16 inches above normal, and I had to use my tractor to feed hay and I just rutted and ate some of my, one of my areas, and it just irritated me to no end. But I still remember when I went to your farm, your operation, and how it was so immensely pugged. So what I did is I took some pictures, and then I started realizing as we got other rains, the ruts started to disappear. So as they got more liquid and went through a stage of liquefaction, started moving like quicksand, you could actually see the ruts starting to disappear. So I found it very fascinating. And you can learn these concepts as you walk in the sand. But if you walk on the edge of the beach where all the water is filling those pores, all of a sudden it starts becoming more turgid, and you can see how that the soil body starts to change. But it's also freezing, and cooling and heating and drying. All of those actions also help. But again, you said it, Ellen, that aggregation, those biotic substances, all these together and living roots make it work very, very incredible. It's an incredible matrix.
Thank you. All right, the next question, and Ray, I'm going to start with you on this one. Compare the biological results from the Haney versus the PLFA versus the chemical or the standard soil test from an analysis by Ward Lab, and be specific on which criteria for the respective tests that you consider most engaging the nutrient needs for corn and soybeans.
Okay, excellent question. Excellent question. First, I always tell people, when you look at the conventional soil test and the Haney test, apples and oranges cannot compare. They're telling you different things. Use both of them, and you use the PLFA. The PLFA I used is, what's the context there? What organisms are present? That's what the basic thing of the PLFA. I want to know the microbial biomass. I want to know what the fungal to bacteria ratios. That's what I use it for. But for me personally, when I'm working with producers, to me, the Haney test is the most important one. Why? First, it uses the concepts of biomimicry. It gives me a snapshot in time what the plant root's going to see when you take that test. So if you take that test in the spring at a temperature of 55 degrees, you get a sample, you send it back to you send it to the lab, they re-wet that soil. Then they use incredibly sophisticated water analyzers and pick up all those molecules, those carbon-based molecules in organic form, nitrogen and organic nitrogen, all those. And they give you credit for those organic nitrogen pools. We have never been able to get credit. The old test did not have the ability to do that. The old test was built on the wrong premise. It was asking wrong questions, and it was using caustic chemistry to force nutrients off the cation exchange capacity. But can we use some of that old soil test to still help us? Yeah. Cation exchange capacity can be useful, percent organic matter. All of them have some purpose. But at the end of the day, that all I feel comfortable with, I'm going to use the Haney test, especially for backing up producers in nitrogen.
David, anything to add to that?
No, I'd agree with a lot of what Ray just said. You know, a couple of things besides just the nitrogen that I look at on the Haney. I really look at the CO2 respiration rate to see how high that is or low, and look at the percent microbially active carbon in that to see how much of that food is being consumed or able to be consumed by the biology. And then on the PLFA side, you know, looking at those numbers can get kind of daunting, and it's always good to see those ratios of what we have in the soil profile, but also to understand that the cyclers of being able to cycle those nutrients, of having those protozoa in there versus not having them in there. A lot of the soils I test across the Midwest, extremely low in those the coyotes, you will, the protozoa and the nematodes. Maybe some bad nematodes, but you know, the other thing is looking at that top number of how much diversity is in there and the total microbial biomass to see how full our soil system is.
Thank you, David. The next question is, what are your viewpoints on feeding plants such as cereal grains, by a biological foliar feed approach versus applying synthetic fertilizers by broadcast or liquid. And they go on to just mention that, you know, there's a number of people like John Kempf, Michael Phillips, Elevate Ag, and others are espousing foliar feeds, and they ask you to be specific on the pros and cons. So, David, you want to start?
You bet. So some of the things that I've seen with the foliar feeding the biologicals and everything, and even just foliar feeding micronutrients, I'll throw that on there as well, because there's a lot of, you know, as you go, you know, a lot of guys try to limit the amount of synthetics, but you're looking at some of these other companies out here that are selling maybe some microbial biostimulants or things are also, you know, and even in the organic markets, you're looking at some micronutrients as well. And so it's really important to understand what that plant really needs. So I typically would do like if I'm thinking about making some type of a foliar treatment, I'll do what's called a plant sap analysis, and that's where I'm looking at the older leaves on the bottom of a plant, comparing them to the young leaves on the top of the plant, and looking at the nutrient mobilization within that plant, and also looking at the cations to anion exchange capacity within the plant, and looking at, you know, typically it's called a Mulder's chart, but it's looking at what nutrient interactions, you know, could be occurring in that plant, because maybe, you know, the tissue sampling or the plant sap might show that boron, for instance, is too low, but our nitrates are really high. Well, if we understand that nitrates actually inhibit the uptake of boron, then why do we have to go apply boron? Just give it a week or two and the nitrates will dissolve and the boron will be right back to where it's going to be. As far as the biological and the biostimulants and stuff like that, you know, there's all kinds of those out on the market. Really be careful of what you're buying, because some of them you're just buying humic acid. By the time you get them, you don't know how long that thing's been sitting in a jug. You don't know what's actually in there. A lot of them might only have maybe 3 to 5 microbes in there, and maybe at most 10 to the 8th power. So when you're comparing putting a quart of that on and hoping that it does some type of good, it's like throwing a 5-gallon bucket of water into the ocean and hoping that you made a difference. You know, you don't know. And there's so much biology on the leaf surface of plants and within the soil profile, that if we just focus on soil health and building those aggregates, focus on using diverse cover crops, we can solve a lot of those issues without needing to apply. Again, that's going back to practices and not principles.
Ray, anything to add to that?
Yeah, I agree with everything that David said. I just want to add that there's a sphere that we hardly ever talk about. It's called the phyllosphere. It's the surface of the leaf. And there's bacteria and also association of fungi that actually create aggregates on the leaf surface. That's been well documented. So these epiphytes, these organisms, can withstand ultraviolet light, but they also help with the hormonal function of the plant. So I would be an advocate only if it was extracts like David Johnson Sioux extract, where you can put it on the surface of the leaf and it helps with disease mitigation, change the hormonal function of the plant. So again, coming back to that whole thing, everything's connected, everything is one. We're finding new, as we're learning more and more science, we're finding out, like David was saying, they're ubiquitous. I'm more about applying the practices and getting off a lot of these inputs and saving. Once you get these system healthy, you don't need any of this. Even Dr. David Johnson will tell you that his compost extract is just a tool and you have to use it in a systems approach. And like David said, one more thing is that you have to be careful. You need to set up your own check strips and that to make sure that these things work. A zero check, and and do your own science. And don't take our word for it, do your own, and don't just depend on any product. And I'm not a huge fan of chemical-based fertilizers. They're a salt, and we need to understand that. And they take huge, copious amounts of energy to make. And farmers are going broke using these products.
Thank you, Ray. Okay, the next question, and David, I think this is for you. It's from one of your pictures that you showed on a slide. Was that buckwheat interseeded in the corn? If not, what was it?
Yeah, I believe that was buckwheat in that 60-inch, yes. There's buckwheat and several other things in there.
Right. Thanks, sir. All right, the next question. Would you consider wildlife like deer as a livestock source for your fields?
Yes, I guess. I was just on a field just along the Illinois River here a week ago, and 40 foot around the perimeter of the field, there's no cover crop growing because the deer absolutely destroyed them. And throughout the whole field, you could see where deer had been chewing on the cover crops and deer droppings throughout there. So yes, it would be just like livestock. The unfortunate thing, trying to manage them to graze effectively, efficiently, the way we'd maybe like to graze them is a little difficult.
Yeah, and I'll add that if Grant and Dawn Brightcroots are on the webinar tonight, I know that they would definitely consider deer as a livestock source because they get overrun by them every winter. Anything else to add from anyone? Okay, the next question. Can you calculate carbon to nitrogen ratio at home, or do you have to take a soil sample and send it to a lab to get the carbon to nitrogen ratio? Ray?
If you're going to want to be accurate about it, you're going to have to send it to a lab. And that's the way I would do that.
Okay. David, anything to add?
I completely agree.
Okay. All right, the next question. Are you, and I think that you is used in general, so would someone practicing these 6 principles use less chemical each year the longer you use cover crops in your rotation?
Absolutely. I've seen so many pictures of people who are using cover crops, and all of a sudden, oh, I'm not having this pigweed issue. Oh, I'm not having these type of weed issues. And being able to suppress out these weeds, either you're suppressing by shading them out, or you figure out that some weeds actually grow because of an excess of or a lack of some nutrient mineral in the soil profile. And when you start growing these cover crops, either suck up these nutrients or bringing some that you're lacking up from deeper in the soil profile, all of a sudden now you're not seeing those weeds. We're seeing that all the time. So yeah, we can start using less and less herbicides.
Yeah, and I would add, we're currently in the middle of doing quite a few written case studies documenting a lot of this. And I can tell you that in 100% of the case studies that we have written up to date, every single one of them has been able to significantly reduce their chemical and synthetic use by using complex cover crops in their rotations. Gabe, you have something to add?
That would be exactly right, as long as they're following the principles.
Yes.
You have to include diversity in there, and you have to include living root in the soil as long as possible throughout the year. You're going to have a difficult time reducing synthetics, including chemical herbicides, if, for instance, you're only in a corn bean rotation, then only using cereal rye, for instance, following corn. You need to get more diversity, and you need to have that living root at all times.
Excellent. Thank you. So the next question, Ray, I'm going to start with you on this. If starting with soils in the low 5 pH before you started using cover crops and or grazing, would you apply lime or just let it adjust upward naturally?
Great question. Depends on the context where you are and how much moisture you get. The example I gave you in South Carolina, the pHs were low. And in fact, they were low. And just 3 years of covers, those plants, those multi-species were able to go down the profile and pick up the calciums and redistribute it and started the healing process. Again, keep in mind, if we're using a lot of these fertilizers that they knock calcium off the exchange site, you got to keep that in mind. You got to also reduce some of these fertilizers that produce a lot of, like ammonium, produce a lot of hydrogen, and they knock calcium off the exchange site. But you also have to be stopping the tillage, and you have to do these other principles that Gabe was talking about if you're going to get and regulate and raise that pH. You just can't just do one thing. You're going to do all these principles to start getting the effect that you want. So it depends on context and how committed you are to pumping carbon. And I start worrying if it starts getting, you start managing for the stresses of the plant, then you add the calcium. But to use calcium to change pH, I wouldn't use that for that concept.
David?
I agree. You know, a lot of times, and you got to be cautious, I guess, on, or not really cautious, but understand that building soil health, if you haven't been using cover crops and you've been using heavy synthetics and a lot of tillage, when you start down the cover crop and this, it takes time. So many people that jump into this sometimes are looking for their saving grace as I'm going to plant this. You know, these guys are saying, you plant this and it's going to work. Guys, it's going to take a couple of years to be able to do this and to get these things to cycle again. But you had to, like Ray said, you had to be committed to it. If you just look at it at a 1 year time, you know, your success is going to be basically the person staring at you back in the mirror. Or
Gotcha.
failure.
The next question, are you checking the fungi levels in your soils as you're starting to use cover crops?
Yeah, I would recommend to anybody starting cover crops to take a PLFA test and take a Haney test so you can baseline where you're started at. And then, you know, either annually or maybe every couple of years, go back out and do the same thing to see how you're progressing.
Excellent. Ray?
Totally agree. He did perfect.
All right, the next question, and this is a producer that's in Kansas on the Nebraska line. So they ask, I've had my cover crop mix of crimson clover, balanza clover, hairy vetch, cereal rye, and winter barley below freezing a couple of times this week, and it's getting snowed on now. If I want to harvest some of this mix for seed, should I be concerned about damage to the rye and barley? The rye is now past joint stage.
So I would say, you know, I'm here in Kansas right now, and if you would wait until probably next week, it looks like the temperature is going to warm back up, and assess your stand at that time, split those stems and look at the head and see, you know, if there's any damage to that, or just right on the bottom side of that head, that little white pith area, see if it's starting to kind of turn off colored, or see if it has an odor to it, you know, and make your determination from there. Unfortunately, sometimes Mother Nature always wins. So we just have to deal with the cards that we're dealt with.
Shane, you're in Kansas. You got anything to add?
I hope not. No, I mean, David made a great point there. I think we need to give a little bit of time and see where everything shakes out at. And like David said, sometimes, you know, Mother Nature's going to win. So just kind of wait and see approach and go check it here in about another 5 to 7 days.
Gabe, anything on your end, Dad?
I would say it's context, and yes, what you need to wait and determine it. But I would say that the money in that crop would be in the hairy vetch and pea seed before the barley or triticale seed. Meaning the value of it, those legumes is much more. So my context would be, okay, if it does harm those grasses, what percentage of the stand is the legumes? Would that still be economically to your advantage to harvest? Because it's rather doubtful that the frost is going to hurt them. And that, right now, hairy vetch, even seed in the dirt, as they call it, uncleaned, is still worth about $1.25 a pound. So take in and consider the whole.
Excellent. All right, the next question, this is a very good question. Would plants that have a high sugar content, like sorghums and specifically like BMR sorghums, contribute more sugar to the soil through roots and therefore stimulate biology more substantially than other crops? David, you want to take a swing at that?
Absolutely. That's my answer. Sorghums, forage sorghums, heck, even milo, and annual ryegrass. It's a high brix content forage that really greatly improves the soil through those root exudates. And one thing I was going to add, and I forgot to talk about it, was as we're building those soil aggregates and everything, and the microbes in the soil are basically releasing CO2 when they're eating, dying, and breeding, whatever, that CO2 leaves the soil profile and is taken up through those plants, through the stomata openings on those plants, and it starts this whole sugar building process over again. So those crops that have high brix crops like forage sorghum, sorghum Sudan grass, annual ryegrass, things like that, that's how they make the soil so mellow and build those aggregates so quickly as well.
Excellent. Ray, anything to add?
No, but I think you did a good job. I just want to add a comment to the last question real quick about that freezing. If the person's doing no-till and covers, the system could be way more forgiving. He may have less damage than he thinks, because these soils will have a huge microbial biomass that will tend to be warmer and could impact the plant in a very positive way. Just a quick comment on that.
Thank you. All right, Ray, I'm going to start with you on this next question. What would your thoughts be on inoculate for peas? Some say it's necessary for the peas to be able to fix nitrogen, but how can we know that the inoculant will be effective? This
is my personal opinion. I think the inoculum is so cheap, I would still do it every year. It's just an assurance. But if you've been using it for a long time, there's different thoughts. If somebody that Gabe he's been planting hairy vetch for a long time, and there's the bacteria in there that are present for creating that inoculation, might not need it, but it's so cheap, and it depends on the context of where you're starting in your soil health journey. If you're just barely starting and you've only been doing it for 3 or 4 or 5 years, it's pennies. I would do it. That's just my opinion, but I think it depends on your context and how long you've been doing this.
David.
Yeah, so different legumes require different rhizobia strain bacteria. So obviously, if you haven't planted those legumes, like if you're just starting out planting peas or even hairy vetch, and that farm or field hasn't seen that crop on it for many years or maybe ever, absolutely inoculate. And like Ray said, it's cheap insurance.
Thank you. All right, the next question. Last year, we ended up with perennial ryegrass bales that we were unable to use because the levels of endophyte toxin in the bales. We were planning on chopping and spreading the bales out onto our land. Do you know if Endophyte toxin has any negative impacts on soil health? David, you want to start with that one?
Well, after listening to Dr. White there a few webinars ago, every plant has an endophyte in it. So whether that's the endophyte that's going to affect anything, you know, positive or not, you know, if I would say there's not going to be an issue to spreading it on anything else. You know, I guess my concern would be if that perennial rye had gone to seed, how was it baled? Was it wet baled? I guess, you know, how might those seeds be active to be germinated or not, would be my own concern if it was on row crop field. But
I'll just add, I've got quite a bit of experience with endophyte infected fescue. And what we have discovered is that as we build a more favorable microbial profile, that we actually see the infectivity levels of the endophyte in the fescue go down, they decrease. And therefore we see less of an impact on our livestock grazing those exact same fescue plants that just, you know, a handful of years earlier, they had significant effects from that endophyte. And now all of a sudden you don't have the vasoconstriction and all of these other issues in the animal's body. So we certainly know that as you improve the overall microbial profile in that soil and you better balance that soil microbial population, that when you have less issues with endophyte toxins in that same soil. Anybody else have anything to add?
Alan, just emphasizes the point that we need to add more diversity instead of trying to grow monocultures, and making sure we have plant diversity in our systems. And that'll help start, as you emphasized, start alleviating a lot of these issues.
Excellent point, Shane. And, you know, and most of the time, people subscribe to that diversity to the plants themselves and just say, well, the animals are diluting the endophyte because they're eating a number of other plant species at the same time, right? And yes, that's partly true. But what we fail to recognize is what we just mentioned. As you add diversity in plant species, you get significantly greater diversity in the soil microbial population, a far better balance. And so it's actually a combination of all of those that is really working for you favorably in that regard. Okay, Ray.
Yes.
Which crops or perennial plants would you plant in severely compacted soils that best meet the soil health principles and context?
Boy, and again, coming back to context, depends where you live and how much moisture you get. But I will tell you this, and I feel incredibly comfortable with the grasses. And to me, the grasses are the most powerful, whether the cereal rye, the barleys, the annual rye. Recently, a landowner gave me a research paper done by Kentucky where they're planting annual rye and how the exudates and the acids coming from that annual rye is actually breaking up the fragile plants and actually getting more depth of soil. In fact, I'll be glad if the person emails me, I'll be glad to share. It was Steve Freeman shared that, and that was research done in Kentucky, the university. And so I'm really excited about what that annual rye can do. So annual rye, cereal rye, any of the grasses with huge fibrous root systems, that's the best way to break up compaction. People brag about radish and those things. No, to me, the grasses are the most powerful.
David, anything to add?
absolutely agree with Ray. The fibrous root systems beat a taproot of a brassica any day.
Absolutely. All right, the next question is, how do we develop a potential for our context?
So I'll start. It's all about understanding what goals you want to achieve. You have to do it for yourself and your farm, not for your neighbor's farm or anybody else. You have to have a really, I guess, get a pad of paper and a pen and sit down and start identifying what you want to achieve on your farm in your lifetime for your generation, your kids' generation, and 7 generations out.
Okay. Next question. In selecting your cover crop mix, are you selecting plants with diverse root depths that exude different exudates to specifically source minerals that may be missing in your A horizon? Ray?
Well, yeah, you try to take as much of all those into play. And one of the things I use as a template is and we teach as an understanding ag, what are your goals and what are your resource issues? And to me, one of the best books that came out was Using Cover Crops Profitably, created by Lasser, and knowing what each tool does and what each plant does, because every plant is absolutely different. It gives a different exudate. The Sudan grasses, all of them, most people don't always realize that some of them can be used to create a natural herbicide, to release sorghum, like Sudan grass. My whole issue is, yeah, you try to bring all the matrix together, but unfortunately, a lot of it's fragmented. You're not going to get a total complete picture. You do the best you can, but there's a little research here, a little research there, a little bit of research there. But I haven't seen, me personally, a document that has all of that, where it takes all the root exudates, which one does that, what it does on the top of the canopy, how it captures energy, what allelopathic material does it leak out the root. I haven't seen that documented, and it should be done, but you try to take some of that into play if you can.
David, anything to add?
Yeah, one thing I tend to look at too is what weeds are growing, or forbs as we'd like to call them, but what plant species are growing, and try to pick a cover crop that might be a kissing cousin to that weed as well, and try to plant something instead of letting nature decide of it. Control it that way. There's some good resources like Weeds and Why They Grow, Weeds Guarding of the Soil, and things like that can give you some idea of what those plants are taking up, or why they grow in that environment as well.
Okay.
So if I can add to that just real quick, like with these challenging times we're dealing with, we need to be looking at things we can take economic gains from.
Yes.
These cover crop mixes, we need to be able to see, how can we take a grazing opportunity from this cover crop? How can we utilize this cover crop for weed suppression, as David alluded to? How do we get from immobilization to mineralization? When do we need that to occur for our next cash crop? So there's many things we need to be looking at. But one of the big things we need to look at with all these cover crops is, how can I find low-hanging fruit, take an economic gain from this while I'm working on getting my soil health to start functioning better?
Excellent. Next question. In trying to regenerate poor soil quickly with a cover crop, do you recommend using a foliar fertilizer with nitrogen to boost growth, photosynthesis, and root exudates? And at what point can you add nitrogen and not shut down nodulation? David, you want to start?
So to work backwards on that, as far as the nodulation, it just depends on the species. Like for instance, I've seen, which I've done some trials with, some starter fertilizers in furrow with soybeans, and significantly had really great above ground biomass growth and really suppressed nodulation at really low rates of nitrogen in furrow. So giving it early on, probably not such a great thing for a legume. As far as you know, it all just depends on what the context of the soil profile is, or if it's compaction or wet or what, you know, to try to get that cover crop to really grow and to establish. Sometimes, yeah, you do have to apply some nitrogen, but you know, some of the things I've seen is some guys that are applying nitrogen to like cereal rye, for instance, because they just can't get the growth out of it. Well, I put a spade in the ground and they have a compaction layer at 2 inches and the roots aren't getting any deeper than 2 inches. And oh yeah, by the way, I have to jump up and down on the spade to get it into the ground at 2 inches. Well, if the roots can't get down any deeper, they're not going to build the aggregate. So we have to understand, you know, sometimes, you know, the necessary evil to break up that incredible hard pan there might be to run a subsoil inline ripper with the no-till points on, so you're not disturbing the above ground much. But what I would do is I'd plant the cover crop, I'd run that across it at an angle, and be that the last time I ever ran any type of implement in there. But you let the roots then get down through that soil profile. You have to be able to get those roots down in there.
Excellent. The next question, as I don't have livestock yet, how would you recommend haying a mainly grass hay field? Should I just limit the number of cuttings?
So yes, limit the amount of cuttings that you do on it. Instead of trying to get 4 cuttings out, maybe only do 2 cuttings on it. And then make sure you've got plenty of growth going into the winter as well.
And I would also recommend that you look at your plant species diversity in that field as well. And if it's fairly low diversity, you may want to look at interseeding some other things to improve the diversity. Okay, here's a really good question for the time we're in right now. With the bottom falling out of the corn market, cattlemen are finding it more economical to feed out with corn at the feedlots. This puts a strain on finding cattlemen and animals to contract graze our cover crops. Any suggestions? Just trying to think of other options in a dry, brittle environment becomes a challenge. David, do you want to start with that?
What about sheep?
There you go.
That could be an option, but you know, sometimes you might have to look outside of your area too, you know, and think about what other markets might be there that somebody might need to fill a niche, you know, for a couple months while they're pastures or something, or you know, growing yet or whatever. You know, I've seen a lot of guys throughout the Midwest already kicking a lot of cows out on perennial pastures when it's really short and not ready to be grazed yet, and kind of wonder why we run out of forages in July. But that's another topic for another date. You know, maybe it's you know, if we can link in with somebody from outside the area to offer some grazing on cover crops instead of just straight up locally, could be an option as well.
Yeah, and that question is from Rickson, and I would add to that right now there's a whole lot of people with cattle in the feedlot that are losing their rear ends, and so they are really wishing right now, even with corn as cheap as it is, that they did not have cattle in the feedlots. So it's certainly not a panacea, even with the cheap corn, to have cattle in a feedlot right now. Gabe, do you have anything to add to this one?
Yeah, I would just suggest look at what we're seeing now with the current pandemic and the interest in grass finish beef. That would certainly be an option for producers with covers, you know, as long as there's not mature corn or something like that in there, to advertise, hey, I have feed available if you're a grass finisher. And that may be an option for them.
Okay. All right, Ray, next question. Any thought or resources on how this concept of pH and nutrient availability relates to human health? Naturally, humans and other mammals are much more complex than plants. Where would I look for information to connect the dots between healthy soil, healthy plants, and human health?
Wow. Oh, Alan, you'd have to say the hardest question for me. And maybe Gabe,
Absolutely.
and maybe Gabe can help here, because Gabe's read more about connecting the dots with human nutrition than I have. I'll just say this real quick. I know there's more and more emerging knowledge coming out about connecting those dots. One of the things in the older literature was, it was very, very well established that the Russians knew. There's a interesting book that I really, one of my favorites, called Soil, Higher Plants and Soil Organisms and Higher Plants, and Greshkinov, and they talked about how adding manures and adding that type of into the soil system, they could actually measure higher concentrations of B12 and in the grain itself. So that's one publication that I would, it's in pieces, and you'll find them a little here, a little there, and it's not, I have not seen it. Oh, there is one book that I would definitely recommend, and when you can go to Gabe and I can pick it up right here quick, that I would recommend, that would help.
Okay, and I would just say that for those interested in this connection from the soil to the plant, to the animal, to the human in terms of our health, we are scheduling right now 2 webinars that will feature 2 of the most prominent experts on this topic. And so we're going to have Fred Provenza and we're going to have Sarah Keogh that will be with us at a later date in May or either late April. We'll have those posted on the website soon, and they are going to specifically address what is being mentioned in this question.
Yes.
Find it, Ray?
Yes, I did. It's called, the book is called Soil Ecology, and it's on, I'm pretty sure it's chapter 3. I apologize, it's downloading the picture on it, but it's called Soil Ecology. And if she sends me, as it's giving me the image of the title here. Another book that I would definitely recommend that's come out this fall, Dr. David Montgomery and his wife are doing extensive research and bringing all those pieces together. And they have done, I mean, those two really dig deeply. And that book will tend to be coming out in the fall. So keep an eye out on how they'll connect those dots from the soil all the way to the plant and to the animal, like Alan was saying. I think they're going to mostly focus on the plant and the soil and to human health. And I'm not sure if they're going to do that with the animal, but I think that you, Ellen, and Understanding Ag may be working on that kind of project, correct?
Yep.
And yep. And
Right.
I will get that, Alan, I will get that. It's having a hard time downloading, but that person that asked me that to email me or to Kathy, and I will send them the copy, title of that book and the chapter where they can find a good resource.
Excellent. Okay, David, this next question is in regards to one of your slides. It says, so the land north of David's line where cover cropping and corn is not feasible, should that land be returned to a diverse prairie and use grazing, and the corn be raised south of your line using the magic of cover crops along with potentially irrigation?
Alan, I'll fill in. David had to step out for a moment. So this gets back to when we talk about context. One of the things we need to look at is historical context. And we're doing a lot of things pushing our farms down a path that historically species would not be grown there. For instance, I was in northern Alberta doing some work last year, and I actually saw fields of soybeans trying to be produced in Alberta. That's out of context. And I'm not going to say that corn does not have a place north of David's red line, because, for instance, if you read Indian Bird Woman's Garden along the Missouri River right here in North Dakota, they were not only growing corn, but they were growing tobacco, the indigenous Native Americans. So what is wrong, though, is we're trying to grow corn when it's cool season. The species, the varieties, I should say, that were being grown here hundreds of years ago were much shorter day varieties. They were 60 to 70 day CRM corn. So we need to put it in the proper context. David, do you want to add anything to that?
I guess I missed that question. I apologize.
It's so the land north of your line in your slide where you had the line and you were talking about cover cropping in corn is not feasible in land north of that line. Should that land be returned to a diverse prairie and grazing, and corn instead be raised south of that line? So that's really the question.
Okay, so that's not, so let me go and address that. So that line isn't that you shouldn't be planting corn up there. What that line was about was like if you were to interseed species like wheat or barley or triticale, those cereal grains, you could do that above that line into corn. So interseeding into V2 or V6 corn, you could seed those cereal grains and get them to survive better than you can south of that line. South of that line, we just have a lot more heat, and it just doesn't allow for those cereal grains to do as well as what like annual ryegrass would do. That's what I meant by that line on that slide.
Okay. All right, the next question is, what is the best time to interseed cover crops and grasses into weed-infested fields or pastures? David, is that something you want to start with?
Go to Ray first, please.
Can you repeat that, Alan?
Yeah, when is the best time to interseed cover crops and grasses into weed-infested fields or pastures?
Okay. Now, I will tell you, it depends on context again. And my experience is with we've been using into permanent grasses fescue. We have used mixes in North Carolina, and we planted a warm season mix into a cool season fescue. But when we did that, we made sure that the main grass that's well established for us, it's fescue, goes into a dormant stage, so that we can allow these mixes that we're using to use them as biological primers to feed the cattle, and maybe get rid of some of the issues with these other forbs. But me personally, I'm always a big fan of doing a good adaptive grazing, and then you change those populations anyway. In other words, as you do better grazing and you do a better job in that type of system, the biology will change and the plant species will change.
Okay, Dave, anything to add to that?
I'd agree with Ray on that.
Okay. Next question. In sandy, low CEC soils, what is the best way to raise the CEC after building organic matter? Is it humates, or so basically how can they raise their CEC?
Just by adding organic matter changes the CEC, the cation exchange capacity. Nothing beats organic matter, not even the clays, to hold on to nutrients It's those super molecules. The best thing you can do is keep planting the covers, and these organisms and all that will create those superbiotic substances that will hold on and change the CEC. So I'm a big advocate of doing it that way.
Okay. All right, the next question. What are your views of livestock grazing plants in their vegetative state versus in their reproductive state? David?
So, when a lot of times when we're grazing on these highly vegetative plants, they're extremely high in protein. So, we have a lot of bypasses going right through them, and we can see that really evident on cattle with how loose they are. And just like you or I would usually feel pretty upset when our bellies are like that. The cattle kind of go through that same thing. Plus, there's some other, compounding effects that can happen and occur when grazing those vegetative plants. When you're looking at reproductive stage plants, I'll use even fescue, for instance. A lot of people fear grazing fescue with their heads out in fear of getting pink eye or something, pink eye issues. And honestly, that's just all a part of your grazing strategy, I guess, or how you're grazing. If you're just doing continuously graze and you kick them out there early on that high lush protein diet, and adjusting that gut pH, we could have actually caused some issues in that cow's. Then the cow to actually cause that pink eye then. And Alan, I know you can go into that in a lot better detail than I can, but I've seen on some areas where we've grazed on fescue again, that using amp grazing on it, we're able to come back to it and be at a stage where it's not real stimmy and hard, as long as we're managing it with the right numbers and everything, and it's never an issue. Ideally, I'd graze in the boot stage if I could, but trying to get animals across all that at one time is difficult. So
Yeah, I'll just add real quick to that yes, that's definitely the ideal stage to be grazing grasses, is in the boot stage, to have the best balance of protein to energy. But the real solution to this is, again, we go back to that plant species diversity. The greater your diversity, the less these types of things become an issue, because you've got plants at many, many different stages of their growth all at the same time, and it's far easier for those livestock to balance their diet. Okay, this next question. How can living roots function when the soil is covered in snow and ice? And Gabe, I'm actually going to go to you to start with that.
Why would you do that now? Come on. They function very well, actually. And there are many papers out there that show, studies that show that plants actually, because of that symbiotic relationship they have with soil biology, they're going to feed that biology to some degree all winter long. So even though here in North Dakota we can freeze a little bit, those plants, though, are still sloughing off root exudates, and biology, some of it is still active. Now, some biology will go into dormancy and it'll sporulate, etc. But it's a very symbiotic type relationship that plants and microbes have, and they know they can't exist without each other. So because of it, those plants are going to make sure they're not going to cut off the dinner to what takes care of them.
Excellent. Okay, the next question, is there a formula for the number of cattle you would use to graze, say, a 2,000-acre grain farm to follow the soil principles? David?
absolutely. So we'd be looking at the size of the cows and their gestation period as well, or lactation period, I'm sorry, to determine what % dry matter they need. And then we'd also be measuring the forages to see how much dry matter is available per acre. And then we can make our paddock size accordingly for either, you know, a daily move, or maybe, you know, if it needs to be a once every 3 day moves or whatever. But we can make our paddock sizes accordingly by understanding how to calculate that.
Okay. All right. And we can provide additional details on that if you want to email Kathy. You know, we can certainly provide the math of that, how to do that. Okay, this next question, and it's from Willie. Willie, I may need a little bit of clarification here. Can you elaborate on the topic that plant organic material residue makes no real contribution to humus, to the humus building process, that this is instead a function of the carbon sequestration process of photosynthate root exudates? So Ray, do you want to start with that?
Yeah. According to the nature and properties of soil, and this has been well documented, that that skin, that residue, people call it residue, the detritosphere, the skin contributes about 25%, 35% into the humic and non-humic. So they break out into percentages. And so a majority goes off into CO2, 8% goes to feed the organisms, but they do contribute some. And like what Willie's saying and alluding to, a majority comes from the roots, exudates, and from the dead microbe carcasses. I think people don't realize this huge microbial biomass and how quickly they reproduce contribute more to the carbon matrix than we've ever imagined. It's not just the scuffing off of the roots in the root system, but the huge populations of the microbial biomass. Recent research shows that 40% of organic matter is dead carcasses of bacteria. So, but the residue, the skin does contribute some of those to those pools.
Hey, David, anything to add?
No, Ray did a good job of that.
Okay. All right, 2 more questions, guys, and then we're going to call it a night. We have had a tremendous number of questions tonight, and we thank you for that. And we do our best to answer everyone that we can. So the next question, is there a certain Brix level that needs to be achieved to begin the process of the plant producing root exudates?
I wouldn't say it's necessarily a Brix level. It's more so as soon as that plant starts to go through the photosynthesis process, it's releasing those root exudates, and even as it's germinating and growing. And you can easily see this just by digging up a seedling and seeing the soil that is aggregated to those roots. So even the little bitty roots off of that seedling, you can see that occurring. You know, that soil doesn't just stick to that root just by chance. You know, it's being stuck to that root due to those root exudates coming out as well.
Ray, anything to add?
No, I think you did a good job on that one.
Okay. All right. So, ladies and gentlemen, we're going to end tonight at that. I do want to thank everyone for participating this evening and for all your tremendous questions. And I want to remind you that we have a number of other webinars scheduled that are coming up. You can go to understandingag.com and go to the webinar tab, and it will give you a list and a description of all of the upcoming webinars. As I mentioned earlier, we're in the process of scheduling webinars to look at that human health connection. Our next marketing webinar will be called Building the House, and we will be going into a much higher level of detail, and we are continuing that marketing series. So we have a host of other webinars that will be coming your way. Continue to go to our website. And also, if you have not subscribed to get our newsletter and all that, for information about upcoming webinars and events and opportunities, be sure and do that as well. So Again, we want to thank everybody for participating this evening, and everybody have a very pleasant evening and a great weekend. Thank you.